And now I would like to welcome our second speaker for the day, Dr. Pallavi Agarwal, who will enlighten us on the pattern of inheritance. Mohit, can you
need to request, we'll request sir to stop sharing.
Yeah, I have done.
And Mohit, you please share Dr. Pallavi Agarwal's slide, please.
Okay, am I audible?
Yeah, Dr. Pallavi Agarwal, just 1 second.
Yeah,
We would like to show your slide first, your CV, and then we'll start, ma'am.
Right.
So Dr. Pallavi Agarwal, she is a professor at the MIT University Institute of Molecular Medicine and Stem Cell Research at Noida. She has done a PhD from the University of Cologne, Germany in genetics and molecular biology. She has over 12 years of post-PhD experience, 4 years of postdoctoral training at Gurdon University of Cambridge, UK. And she has been awarded with DPT Biocare Women Scientist Extramural Grant. Her research is exhaustive, and she's done it in understanding the role of genetic and epigenetic regulation, gene amplification in ovarian tumorigenesis, drug resistance, radio resistance in gynecological cancer. That's extremely exhaustive and impressive, ma'am. Welcome to our program, and please enlighten us on the pattern of inheritance.
Thank you very much. So if you want, I can share the slides myself. I sent you just in case, actually.
Yes, ma'am, you can share. Just give us a second. Mohit will stop sharing and you can start sharing. Mohit?
Yes, ma'am, you can share your screen.
Okay. Wait a moment. Mohit, maybe you can share. That will be faster.
Okay, ma'am, sharing.
Yeah. I'll just ask you to change the slide as I move. Yeah, could you do it?
I'm sharing, just give me a minute.
Yeah. So meanwhile, I would really like to thank the organizers for giving this opportunity. And I think this one-day course is an extremely good thing to do because genetics is something which is base of us as how we grow, how we transmit our genes. All this information are so important, and it will give a comprehensive understanding to the audience here. And I'm really thankful to Dr. Pankaj Talwar, Dr. Nisha, and everyone for inviting me. So maybe you can do a slide share. Right. So can I start, Dr. Nisha?
Ma'am, please do.
Yes, Dr. Pallavi, yes.
Yeah, thank you. I'm Dr. Pallavi Agarwal. I'm a professor at MIT Institute of Molecular Medicine Stem Cell Research. And I'm happy to present this topic, which is patterns of inheritance. Now, you got a very comprehensive understanding of DNA from Professor Biswas, who is my colleague, as well as the director of the institute. And I think this will really take you to the next step here with understanding patterns of inheritance, because what are we talking about inheritance of? We are actually talking about the inheritance of genes, and these genes are residing on that DNA for which the structure and function of the DNA, which you just went through in the previous talk. So we'll understand a bit on actually us, basically. Can you go on the next slide, please? Okay, so our body is made up of trillions of cells, and each of our cell has got nucleus, and each nucleus has the genetic material. This genetic material is found in the form of chromosomes, right? And these chromosomes are basically made up of DNA, which you just looked at, the ATGCs, which are the long strands of DNA, but they are actually found as independent chromosomes in us. And if we talk about humans, we have 46 chromosomes or 23 pairs of chromosomes, as what we say. And each chromosome has got they are present in pairs. That means we have a homologous chromosome for each chromosome. So if we talk about our chromosome, we have chromosome 1 to 22, and we have one pair of sex chromosome, right? And in our chromosomes, we have at specific loci, we have genes. Now, what are these genes? Genes are actually the functional unit of genetic inheritance, which can actually code for a functional protein. And these proteins determine the traits or the phenotypes that we look at. Like, how do I look, the color of my hair, the color of my skin, the color of my eyes, all these phenotypes, physically what we see is actually been determined by the genes, right? And these genes, we get it from our parents, from both the parents, from the father as well as from the mother, okay? So when we talk about traits, so these are determined by the genes which are residing on the chromosomes, and a gene is a segment of DNA that determines a trait. Next, please. So when I'm talking about traits, it's basically all the different phenotypes we talked about. So from species, from animals to any organisms, they all have different traits, and all these traits are regulated by genes. Now here, what you see at the bottom is basically some of the specific traits which are also genetically influenced, basically. So the rolling of the tongue or the dimple that we see in an individual. So all different kind of facial characteristics that we talk about, or the cleft or basically the chin structure or the curly hair. Every of this is what we call as traits or the bodily characteristics, the physical characteristics. And all these traits are basically passed on from parents to offspring, and which is what we call as genetic inheritance. All right. So that's what we are trying to understand, that how the genetics work and how these patterns are inherited from parents to offsprings. Next, please. So from where this concept of genetic inheritance came? I mean, okay, genes were, if you talk, just looked at the previous talk, the real structure and major functions of DNA was only understood in 1953 when the DNA was, structure was revealed, even though people did various experiments where they determined whether it's the RNA, protein or gene, what is the actual reason for genetic inheritance. But the concept of genetic inheritance was long way before being provided by Mendel, who basically is actually he was an Austrian monk, and he did not have enough money, so that's why instead of going and continuing his education, he basically took up gardening in his garden of monastery, and there he was very intrigued by the pea plant, and he was continuously growing it. And what he found, that there were many traits in the pea plant, whether we talk about, say, the color of the pea, the pod, the number of seeds in the pod, or say the color of the flower, all these had some distinct features, and they can be counted, they can be understood, and they can be calculated mathematically. And based on that, he actually studied these 7 different traits in pea by performing mating crossing in the pea plant. And so I think one thing which we all know about genetics, that and especially about Mendelian genetics, that Mendel was very, very lucky that all the traits that he picked were actually pure breeding. That means there was no mixing, there was no exceptions, and he could actually very nicely show that how the genetics work, and based on which the three fundamental laws of inheritance was been provided. And because of his contributions, he's been known as father of genetics. And this all was long back in the 19th century when he did all these experiments. But largely his all contribution went unnoticed for a long time. It was only in 1900 when other scientists saw the similar experiments, did similar things, and then they found that it was Mendel who already established all these aspects long back. Right, next please. So, so again, before we go into the specifics of the laws, let's just look at the homologous chromosomes. So here what you see is actually the karyogram of the chromosomes, and in this karyogram, what you see is a homologous chromosome. Now, each homologous chromosome has two sister chromatids. So the homologous chromosomes, the sister chromatids are identical, while the homologous chromosome, they are basically duplicate, but one is coming from the father and one is coming from the mother. So even though the genes are residing on the same locations, but they exist in a different pair because they are two alleles for each gene, right? So what we understand that the homologous pairs come one from the female parent and one from the male parent, and thus all chromosomes, they come in homologous pairs, and genes also come in pairs. Next please. So let's go to the first law which Mendel gave, that was the law of dominance. Now, with all his studies, he could show that basically the genes, or which the factors, that's what he called, the traits are determined by factors, and these factors come in two pair. So they come as either dominant or recessive. Now, what does that mean? That if the dominant allele is present, it will mask the expression of the recessive allele. So the presence of dominant is masking the recessive. Let's take an example here. If this is a pair of chromosome and the homologous chromosome, and one is coming from the father, another one coming from the mother,
I think there is a network problem, Mohit. Is it there is a network problem from end of the speaker?
Yes, ma'am.
Yes, ma'am. Yes, ma'am.
So let's please wait. Yeah.
Yeah, that's it. So I had a word with Dr. Pallavi. She had some network issue. She's trying to rejoin. Please bear with us for some time. Till then, if anybody has any questions, they can ask or something. She's rejoining. Just wait for 1 minute, please.
Till she rejoins, if the audience has any questions, and if Dr. Subrajit Biswas is still with us, you could please ask some questions in the meanwhile.
yeah, am I audible?
Yes, you are, Dr.
Yeah.
Pallavi.
Okay.
I'm
Good,
extremely
ma'am.
sorry for this.
No.
Please go to the next slide, please. Yeah, next slide. Right. So we were talking about law of dominance. What we understood that there are the genes occur in two alleles. One can be dominant, one can be recessive. And it is a combination of those two alleles which we get from our parents that will eventually say whether that phenotype will express itself in a dominant way or in a recessive way. Now, that takes me to the second law, which is known as law of segregation. Now, we all know that in a zygote, basically the germ cells from the parents, that means the sperm and the ova, they together make the zygote, right? And this sperm and ova, actually, they are not diploid in nature. They are haploid in nature because they undergo the process of meiosis. So here up there, you can see that the germ, so basically the spermatozoa or say the ovum, it is basically been formed because there is meiosis 1 and meiosis 2. In meiosis 2, the germ cells are haploid in nature, and these haploid cells have equal chances of meeting the other parent's haploid cell to make the diploid zygote. So that is what is known as law of segregation, that the pairs of alleles or the gene variants and their corresponding traits, they separate into the reproductive cells, and then they have equal chance of recombining. And that is the reason there are equal probability of how the germ cell combine and the offspring will be produced. That is why we say that what is the probability of the offspring of having a certain disease. So we always do a cross and look at that, okay, the probability could be 25%, 50%, and so on. Now, let's go to the third law, which is basically talking about just not about one gene, it's talking about two genes. And this is specifically for genes which are either present on two different chromosomes, or they are actually so far apart that they are not influencing each other. So again, as I told you, Mendel was very lucky that all the genes and all the traits that he picked up were actually locating on different chromosomes. And that is why he can come up with this law, which is law of independent assortment. So if you look at again, you see that if there are two pair of genes we are talking about, they do not influence the expression of each other, because the expression comes from the genes and the alleles. That means the alleles can segregate independently for two different genes as well, and they do not influence the expression of each other. Now, all this actually is quite basic understanding, which we have developed quite early in our studies. You all must have studied, so I will not really go much deeper into that. Let's go to the next step, which is understanding that based on these laws, how different genetic diseases are inherited and what are the different patterns of inheritance. Next, please. Right. So we have understood what is a dominant allele, what is a recessive allele. Now, how do we express them? How do we write them, basically? So there are two terms which is known as genotype and phenotype. So phenotype is all what we physically see, the morphological structure or the morphological physical observation. That is known as the phenotype. Now, genotype is basically based on the allele which is expressing for a particular gene. So in case, say here, capital D is representing a homozygous dominant genotype. Capital D, small d is representing a heterozygous phenotype. I wrote dominant there because, as I again reiterate, the dominant will mask the recessive. So the phenotypically, this heterozygous genotype will be dominant phenotype. And lastly, you have homozygous recessive. So here, in case of a disease which is recessive in nature, if it is having a small d allele, then it will be expressing the disease. So this is how we write the genotypes. And all that, because of this genotype, the traits that we observe, which might be a dominant trait or a recessive trait, is what we call as a phenotype. Next, please. Right. So let's talk about the pattern. So in case a disease is following the Mendelian pattern of inheritance, the pattern can be determined based on two major things. The first is whether the disease allele is present on autosomes or on the X chromosome or Y chromosome, say. So whether that particular gene is residing on chromosome 1 to 22 or present is on the X or the Y chromosome. The first thing is what we see there. The second is whether the disease allele is present in a dominant form or in a recessive form. What does it mean? That the disease will express itself if it is dominant in nature or it is recessive in nature. So that's how, when we understand both these aspects, we'll be able to understand what kind of pattern is being followed for this particular disease. Now, based on these understanding, the different patterns that we see are autosomal dominant inheritance, autosomal recessive inheritance, X-linked dominant inheritance, X-linked recessive inheritance, and Y-linked diseases. So we'll go through the one by one with one example to make it simpler for you. Next, please. Right. So let's talk about autosomal dominant inheritance. So autosomal would mean that this particular diseases the gene is residing on an autosome. That means from chromosome 1 to 22, on any of these chromosomes, this gene is residing. It is dominant in nature. That means if it is present in a heterozygous form or if it is present in a dominant heterozygous dominant form, in both cases, the disease will express itself. Now, there are some observations or features that we see when an individual is having an autosomal dominant inheritance. We will find that the affected person usually has at least one affected parent. So this is actually very straightforward. In case a disease is dominant, there is no skipping of generation. That means every generation will have the disease. So that's why we always find that an affected parent is causing the disease in the offspring. The next thing is it affects either gender. So this is again a feature for autosomal diseases, that it affects male and female equally. There is no gender bias in such cases. And it is transmitted by either gender. That means both male and female can transmit the disease. Now here is listed some of the diseases that follow autosomal dominant inheritance, such as Huntington disease, achondroplasia, familial hypercholesterolemia, and others. Next, please. So just with an example here, let's have a look. Here, the father is having the autosomal dominant disease. He is affected, and you see that only one of the allele is affected. That means one of the allele is dominant there. Now, when the cross is made, the father can transmit it both offspring. So there is a 50% chance that the child might be affected. Could be a male or a female, it doesn't matter. But because the disease is actually lying on the autosomes, right? I hope with this example, you understand this disease pattern better. Next, please. So the next is autosomal recessive inheritance. Now, again, let's go break it and understand. Autosomal, that means the gene is residing on autosomes, chromosome 1 to 22. It is recessive. That means for the disease to express, both the alleles should be recessive, right? So in such cases, we find that affected people are usually born to unaffected parent. Now, how can that be? This is because both the parents could be heterozygous in nature. They are asymptomatic carriers and they do not express the disease. But when the parent alleles are combining, then the possibility of a homozygous recessive is there, which will express the disease. So we call this as skipping of generation, that in case of recessive diseases, the skipping of generation is observed. Now, this is very common in cases when marriages are happening in the same families, when the marriages happen in cousins or in the similar family, because there are chances there is a predisposition of a genetic disease in the family. So the autosomal recessive inheritance diseases are cystic fibrosis, sickle cell anemia, Tay-Sachs disease, and many other are being reported. Could you, next slide, please? So this is an example for sickle cell trait. You, again, as I told you, both parents are heterozygous, so they are asymptomatic, and there is a chance of 25% that the individual with sickle cell anemia will appear because both the recessive allele recombine, and finally progeny might be affected. Next, please. So we talked about autosomal diseases, let's talk about sex-linked diseases or X-linked diseases. So some of the traits might be present on the X chromosome or some might be present on Y chromosome. Okay, but but if we talk about the size of the chromosome, X chromosome is much larger, the Y chromosome is much shorter. So most of the diseases that we look at are X chromosome diseases, while Y chromosome diseases are much lower in number, but also because Y chromosome only exists in males, the transmission is majorly from male to male transmission. We'll just talk about that in a while. So if we talk about inheritance patterns that are sex-linked, they are, one is X or Y-linked, the other is sex-limited, and the other is sex-influenced. We'll talk about them one by one. Next, please. So in case of X-linked dominant inheritance patterns, what do we understand? The gene that we are talking about is residing on X chromosome, first thing. Secondly, it is present in a dominant form. That means if there is only one X and if that has a dominant allele, that means the disease will express. Now, let's just talk about, in this case, because this is a X-linked disease, we do not talk about DD or AA or those things. We talk about X combination of XX and XY, right? So we see that a female has two X chromosome while a male has only a single X chromosome. Okay, so in this case, we find that X-linked dominant inheritance diseases are more common in females than males. Now, this could be intriguing why there is a gender bias. This is because the X chromosome in a female comes from both the parents. It comes from father as well as from the mother. Okay, so it has, say, chance from both, 2 times chance basically, both from the father and the mother. But in case of a male, the X chromosome only come from the mother, while the Y chromosome comes from the father. So in that cases, we find that if it is a 2x chance for a female to get a X-linked dominant inheritance disease, it is only 1x chance for a male. So there is a gender bias. So when we make pedigrees, we can very clearly look at, if we see that there is a gender bias towards female, that would tell us that it might be a X-linked dominant inheritance disease.
Dr.
Now,
Pallavi, ma'am, a gentle reminder, ma'am.
Yeah.
We're already
Okay,
running a little late, but ma'am, you have 5 minutes because then we go into a short break.
Right. Okay. Thank you. So
Yes,
I'll
ma'am.
just wrap up quickly. So what we find that there is a because these are dominant, so therefore this basically there is no skipping of generation. We will find the disease in all generation, and for an affected male, all the daughters, but none of the son will be affected. And these are some listed here are some of the diseases that follow X-linked dominant inheritance patterns. Next, please. So here, just to highlight on here, in this case, we find that the father is affected on the left-hand side, and in the right-hand side, we have mother who is affected. And you can clearly see that in case of a affected father, it's the daughter which gets affected with the disease. And in case of a affected mother, it's the equal chance of son and the daughter to get affected, depending on, so 50% of chance of the progeny to be affected. Next, please. So the other is X-linked recessive inheritance. Here again, we see a gender bias, but towards males. We find that the X-linked recessive inheritance is because of the presence of the recessive allele. And if even one of the X chromosome from the mother is having the recessive allele, it has much higher chance for the male to get it from the mother, while the other X comes from the father in case of the female. So in this case, in X-linked recessive inheritance, we get a bias towards male. And it is recessive in nature, so we'll be able to see skipping of generation. It is possible that there are the mother is normally an asymptomatic carrier and may have affected male relatives. So some of the examples for X-linked recessive are DMD, SCID, color blindness, hemophilia, which are well-known diseases which follow X-linked recessive inheritance. Next, please. Again, some example here. Here we find that the father is affected with the recessive allele or the mother is an unaffected carrier. So in this case, both the parents are unaffected, but if it from the mother it passes on to the son, then the son will get affected while the daughter will remain a carrier. So in that case, only 25% of chance for the disease to express. Next, please. So now I come to Y-linked inheritance. The pattern is very clear. The Y chromosome moves only from father to son and so on. So it is a complete male inheritance. And some of the diseases that we see are like azoospermia, where we see mutation in multiple genes. Retinitis pigmentosa, PRY gene is affected in Y chromosome, and testicular disorders, SRY gene is affected. And this is a pedigree which shows the Y-linked inheritance. Next, please. So initially I also talked about something called sex-limited traits, which means that these traits are visible only within one sex. The reason is, while the gene is present in both the sexes, but it will be expressed in only one gender, which is majorly determined by the hormones. So what we find with sex-limited traits, that in the pedigree, we find the trait only appearing in one sex. One of the example is the barred coloring in the chicken, which is found only in the roosters, and also milk production in mammals. This is again, even though the gene is present in autosomal genes, but this is sex-limited trait. It is only limited to the females because of the hormones. Next, please. So this is my last slide, basically. So we talk about a sex influence trait. The difference here is that it will basically be in, again, the genes are present in both, but they may be also expressed in the both depending on how the hormonal status is. So for example, premature baldness, which is actually autosomal dominant trait and which is found very commonly in the males, but it is the female hormones actually suppresses it. But after the menopause, when the hormone levels are reduced, this particular disease might appear. So that is known as sex-influenced traits. Yeah. Okay. So with this, next slide, please. Thank you. Okay. So I thank all the organizers, and I hope it was intriguing and interesting for you. Thank you for your time.
Absolutely, ma'am. It indeed, like, we have a lot of confusion prevailing around the autosomal recessive, the sex-linked recessive dominant, inheritance. So that was really enlightening. Thank you so much. And our audience, if you have any questions, please go ahead and ask our two speakers. They are there for you. And within this time, up till 10, also we'll be taking a short break where you can all possibly go and grab yourselves a glass of water or a coffee. But before that, if any questions, please go ahead and ask.
Yeah, I'm Dr. Preeti Jindal from Chandigarh. Can I ask a question, please?
Please go ahead, please.
Okay, so I have a patient with who has been diagnosed with polycystic kidneys, a girl, female. So in her case, what test you would recommend, or what type of inheritance you would recommend that I should go ahead with her husband to make sure that her progeny does not have polycystic kidneys?
So Dr. Pallavi, please guide her with that.
Yeah. So, so is the disease already known to be undergoing any inheritance?
No, it is just an incidental diagnosis. On her ultrasound abdomen, she came with some other gynae problem. We got an ultrasound abdomen done, and it was found that she has multiple cysts in the kidney.
Right.
So the nephrologist says that, ma'am, that you can go for the genetic mapping for the polycystic kidney disease. So how should I go about it?
Yeah. So in such cases, basically, either one has to, because as such, it is not very well clear that polycystic kidney is a inherited disease, but there might be, so there is something called as there's a mapping of Mendelian inheritance diseases.
Pedigree chart, probably we have to look into the family history.
Can I answer this, please?
Yes, Dr. Nisha, please.
Yeah, hi. So, hi, Dr. Preeti. This is Dr. Nisha. So, basically, PKD, some 90% of the times it is an autosomal dominant thing. Okay, so you will have
Right.
to check, you will have to get a consultation with the geneticist, and they will do the pedigree charting, take the samples from the one of the parent usually has it, and there is a 50% chance of transmitting the disease to the child. So you need to get a consultation done with the geneticist and get the genetic analysis done of the parent as well as the children.
All right. Okay. Thank you. Thank
And
you.
other thing, just to add that you can go for maybe the next generation sequencing, because if it is a new mutation or it is a de novo disease, then those can be correlated with the existing data. So that is another thing that could be done.
So
Okay.
anybody
All
wants to understand how pattern of inheritance works, how it is inherited from one parent to other to the child, and what is generation skipping, or anybody wants to have any questions about the DNA structure and all, you are most welcome to ask. Our both the speakers are here. Otherwise, we take a break for 15 minutes now.
Yes,
Right,
ma'am.
Dr.
Then
Vishnu?
we connect by 10 o'clock, ma'am, and we
Yeah,
start
10
with your
o'clock we
call
will be
then.
here
Yes.
again.
Yes.
Okay. Okay.
Thank you so
Thank
much.
you, Dr. Pallavi. That was excellent. Thank you.
Thank you so much. Thank you.
Thank you so much. Thank you.
Thank you.